Hi everyone, my name is Nousha Afshari. I work in the Spartan Physics Lab at LSU with Dr. Jeffrey Chancellor. Today I'll be discussing the evaluation of radiobiological models. The purpose of radiobiological models is to describe cellular effects from ionizing radiation. The founding scientists who developed the theories based their models on the same assumption, that nuclear DNA is the primary target and essentially the only target of interest for ionizing radiation. Their assumptions are based on an experiment. This experiment involved irradiating a cell composed of cytoplasm and DNA in vitro, or in a petri dish, and from this experiment it was found that of the two structures, DNA is the more radiosensitive. So now looking at the images on the slide, the image on the left is prokaryote, the image on the right is eukaryote. The prokaryote cell is more similar to the cell described in the experiment. But the eukaryote cell, which is also called a mammalian cell, is much more complicated. There are more organelles, and the process of this cell to maintain homeostasis is more complex. So as a result, models that were developed were built on the assumption that the simplistic prokaryote cell and the complex eukaryote cells are essentially the same. Current models view DNA breaks, repair, misrepair, and cell survival through the lens of radiotherapy treatments. But these models cannot reconcile how changes in functionality at the subcellular level corresponds to whole body effects. So, for example, stochastic effects like cancer occurrence and eradication can be evaluated from these models, but radiation side effects cannot. And that's because radiosensitivity depends on more than just the presence of nuclear DNA. It depends on the cell type, the microenvironment, the radiation type, and the timing of the radiation during the cell cycle. So how functionality of subcellular structures is affected by ionizing radiation needs to be taken into account. So starting at the subcellular level and looking at how that dysfunction of a cell and how that affects how it communicates with other cells corresponds and evolves into whole body effects. And that concludes my presentation. Thank you.